Okay, so it's my great pleasure to introduce our next speaker, Dr. Emma Lundberg. Emma is associate professor at Stanford University, and she's also professor at KTH, the Royal Institute of Technology in Stockholm. Her research focuses on mapping the spatial organization of the human proteome. And among other things, her lab is creating the cell atlas within the Human Protein Atlas database. And today, she will talk about how to decode human cell architecture. So please join me in welcoming Emma.
Thank you for having me, even though I don't work with metabolism, so it's a bit, I guess, daunting to be here. But I will talk about proteins, and I hope you enjoy that anyway. So my lab is very focused on understanding the subcellular organization of cells to measure and model the spatiotemporal organization of the proteome with the goal to understand how variations in protein distribution can control cellular function or lead to disease. And first of all, to map where proteins are in cells, something we've been doing for a long time. And I will just give a basic intro to what we have in the Human Protein Atlas. So it's a Swedish program that I hope many of you are using, hopefully on a, daily, weekly basis, where we've generated antibodies for the majority of human proteins. And we've validated these antibodies extensively. And I'm happy to talk about that in the break if anyone is interested. But basically what we have is a proteome-wide library of binders that we can deploy in different assays to measure where proteins are expressed, for example, at the cellular or subcellular level or in biofluids. The key data type that we have in the atlas is images. It's actually one of the largest systematically acquired image collections in the world. So we have immunohistochemical images. Let's see if I can point to the left here. That shows protein expression in different cell types across the human body. And then we have the confocal microscope images that show the subcellular distribution of proteins. So this is what my lab has been creating over the years. And I will just show one image and keep this in mind for later in the talk. So we do stain the cell with some reference markers. DOPI for the nucleus, an antibody-based marker for microtubules, and the endoplasmic reticulum to get an idea of what the cell looks like the nose and the eyes and the ears of the cell. And then on top of this, we use our in-house generated antibodies one at a time to study all human proteins. So this is CD44. It's on the plasma membrane of this cell. If an image looked like this, we'd look at this green pattern. It's the protein of interest, and we'll annotate and say this protein is in actin filaments. So it's a pattern recognition game, basically. This protein is obviously in centrosomes. You can see the two dots here. So that's what we've been doing systematically for a very long time. And we published the Cell Atlas quite many years back now. And by now, we've localized a bit over 14,000 human proteins in cells. So that's probably most of the proteins that you will find intracellularly in cells. And I should also say that the first time, it sounds very, very basic, and it is very basic, but the first time when we published this paper, half of these proteins had no evidence of where they were in cells. So even though it's very basic, it's, I don't know, people hadn't really studied it. So the two aspects that we find particularly interesting would be patterns like this. To the left, you can see a protein that is in two different compartments in the cell. So you can see that this protein is on the plasma membrane and in the nucleus. And the cells have been compartmentalized to allow different kind of reactions to take place in different parts of the cell. So it might be so that this protein is involved in different biological functions in these places. And then we're also quite interested in proteins like this that show heterogeneity within this homogeneous cell population. So this is actually a cell cycle protein, and we know it's highly expressed in some cells and lowly in others, for example. Most often, we see a combination of the two, so it looks something like this. And this is enolase 1. It's a very well-studied protein, and it has many known functions. So for example, here, we know that the glycolytic function happens in the cytosol. It's binding DNA and regulating gene expression in the nucleus, and it functions as a plasminogen receptor on the cell surface. So I think it's quite fascinating to think about that these cells that are more or less identical show very different functional capacities when you look at just the localization of enolase 1 alone and the expression level, the brightness here. But we're also a systems biology lab, so we're not interested in enolase 1. We're interested in this phenomena from a systems perspective. So when we look at all our data, we can see that 59% of human proteins localize to multiple compartments. And this has also been shown for other species too, so it seems to be just above 50%. And these proteins, of course, they might perform the same function in different compartments, but they might have moonlighting functions and give rise to pleiotropy. And in terms of dynamics, what we can see in our images, about 21% of proteins show signs of single-cell heterogeneity. I think this is an underestimate because this is still just cell lines grown under kind of steady conditions, no perturbations, but this is what we see in our data. So over the past couple of years, we worked a lot to try to understand this variability that we're seeing in our images. And the obvious one to start with was cell cycle. We assumed that everything would probably just map to the cell cycle. And I'm just going to show you one slide. This was published a few years back. We mapped all these proteins that showed variability to cells expressing cell cycle markers, so we could reconstruct the cell cycle pseudotime here, and we could show RNA expression and protein expression. And these are two examples of two new cell cycle proteins that we've found, and you can see that they cycle at the protein level, but they're stable at the RNA level. So in total in this study, we were able to identify 300 new cell cycle proteins, and most of them showed patterns like this. So stable at the RNA level, that's probably why they were never found before, but cycling at the protein level. So that was a very nice study in itself, but what was most interesting here is that our initial hypothesis was wrong. We could not explain the majority of the variations by the cell cycle. So 2/3 of the variable proteins that we saw could not be explained by the cell cycle. So we did some more digging, and this is the current count. A bit more than 3,000 human proteins in our data show single-cell variability. 539, we've been able to determine that they're cell cycle dependent. 1,180 are independent of the cell cycle, and many of those are metabolic enzymes. And that made us very curious. So we took a step back and said, okay, let's just systematically assess the spatiotemporal distribution of human metabolic enzymes in single human cells. So again, if you look at the image in the background here, I'll tell you which protein it is later. You can think about if you know which one it is. It's an interesting example. You can see that it's located to vesicles and the endoplasmic reticulum in some cells, but then in some cells it's exclusively in the nucleus. So this is a quite dramatic biological perturbation, because all of a sudden it's not present at all in the vesicles. But I'll get back to that. So in total, when we look at metabolic enzymes, 40% of all metabolic proteins show single-cell variability in cultivated cell lines. And we see this enrichment for enzymes involved in nucleotide metabolism and central metabolism. So we don't really see it in kind of PTM regulation for those type of enzymes. And often, as an image here, of course, if we look at two enzymes that are involved in the TCA cycle and they're high expression in some cells and low in others, we do see a high correlation. So we've done all the sanity checks of things we know should be co-expressed, for example, but there's a lot of surprises in there too. So again, this is HMG-CS1, an enzyme involved in cholesterol biosynthesis. Again, this image looks kind of similar to the previous one. We have differences in intensity and also differences in localization. Sometimes it's in the nucleus, sometimes it's in the cytosol, sometimes both. And the canonical function happens in the cytosol. I think the nuclear function is still not resolved. So this is where using antibodies, you should be very aware of that. The most common cross-reactivity will be to the nucleus, because the nucleus is sticky. So we're always a bit worried about that. And reviewers are also always worried about that. So the first thing that we did here is that we wanted to see, is this really, like, can a single cell really lead to all of these different phenotypes? So if we tag this gene with GFP and take a single cell and let it grow to a clone, will we still see the same variability? And this is something we did with Manu Leonetti at the Chan Zuckerberg Biohub in San Francisco in HEK cells, and this is the results that we got. So clearly we can see the same phenotypes, the nuclear localization, the cytosolic, the mixture, the difference in intensity. And that was the case for the nearly 80 enzymes that we tested. For most of them, we could reproduce this variability, and it seems like a single cell can give rise to this well-mixed population of different states, if you will. But then, in contrast to the cell cycle, cell cycle was easy because we had this pseudo-time we could map the data to. But here, we don't know are they cycling, how are they going in and out of states. We don't have any pseudo-time to map it to, and it was very hard to do live cell imaging as well. So we ended up with some, this is a boring plot, but it's an important one, so I'll walk you through it. Just looking at populations. So we're looking at all proteins, all proteins, all variable proteins that are cell cycle dependent, and all variable proteins that are not cell cycle dependent. And then we look at a population of cells, and we look at the Gini index, a measure of the inequality of the expression level in this population of cells. And then we look at that at the RNA level and at the protein level. And what we can see here is that for all proteins, the Gini index is very similar for RNA and protein in general. If we look at the cell cycle proteins, we see that the Gini index, the inequality, is slightly higher at the protein level than at the RNA level. And I think this is pretty well accepted. And if we look at the variable enzymes that are not cell cycle dependent, we see an even greater gap. So the protein variability is even higher, and the RNA variability is even lower. So even though we can't necessarily say that this hints at post-transcriptional regulation, we do believe that these localization changes and abundance changes are mainly, I guess, post-translational events. But that remains to be proven. But that could also explain why very few single-cell sequencing studies have picked up on this, except for some beautiful work in the sphingolipid space, I would say. So far, we've shown that there's a lot of single-cell variability for metabolic enzymes. We know that it's a dynamic expression, and it seems to be post-translationally regulated. So then if we think about this other aspect, not time, but space, if we look at multilocalization, it's very common. 56% of metabolic enzymes show multilocalization, and we see this for many different types of metabolism as well. So here you can see for OXPHOS or cholesterol synthesis, these are the proteins that are in the cytosol, and many of them are also in the nucleus, for example. So we see these very complex patterns of multilocalization for many, many types of enzymes. And this is where we find protein-protein interaction data to be incredibly valuable. I wish people would generate more such data at scale. So if we go back to that first image that I showed you of this enzyme, it's actually P4HA2, so it's involved in collagen synthesis in vesicles. The function in the nucleus has not been described, but when we pull together all the protein-protein interaction data we can find, we get this little network plot, and then we see two clusters. One cluster is this cluster, and these are all the collagen components, and of course they localize in a very similar kind of vesicular pattern. But we also see this pattern here, this cluster, and these proteins have a very similar granular nuclear pattern as this cell. And it turns out that all of these proteins are involved in RNA processing. So based on this, we would hypothesize that maybe this enzyme has a secondary function involved in RNA processing in the nucleus. But of course, it takes a lot of time to do this for every single enzyme, so we want to figure out a way to do this at scale. And then I want to show one more figure. It's a bit busy, but the reviewer asked us to multiplex all these enzymes. And of course, we can't multiplex 3,000 antibodies, but we did a pretty massive experiments where we multiplexed 135 enzymes in smaller batches together with some reference enzymes, and we were able to use some modeling approaches to bring it all into one space. But what I do want to point out here is that basically we can show that these metabolic states, if you will, or these enzyme states, the cells have a continuum of these states. It's not only two discrete states, for example, that they go in and out of, and it's not necessarily a cycle either, but there's a pretty complex continuum. So this is something that we're very excited on following up on. So at this point, this is what I have on metabolism. We took this classic view of the kind of biochemical reactions that happen in a cell and just colored the different reactions by whether the enzyme catalyzing that reaction showed variability in a cell or not. So it's pretty much all over the place. I think if anyone here is working with metabolic modeling, it would be very cool to see metabolic models built on single-cell proteomic data instead, and maybe even spatially resolved to see if it might change the way we think of metabolic states and dynamic metabolic states. So our current conclusion hypothesis is that metabolic enzyme spatial-temporal partitioning establishes non-genetic dynamic metabolic states. And right now we're doing some pretty large perturbation studies, so we can see how we can shift cells in and out of this kind of different populations in this continuum and see how protein localization and enzyme localization changes. If you want to explore this data, there's a preprint and there's also some sections on the Protein Atlas. So all the data is already published and we've summarized it in there into landing pages on the Protein Atlas as well. Okay, and with that, I wanted to talk a little bit about the most recent addition we had to the Cell Atlas of the Protein Atlas, which is an organelle called the primary cilium. And it is like an antenna, right, that sticks out of cells, so in non-dividing cells. It's present on most cell types in the human body, but not all. And they're very difficult to study because they are maybe 2,000-3,000 times smaller than a cell, and you can't really isolate them. But they're, of course, they function as sensors, and they're very important during development. And most of our knowledge of the cilia comes from studies of ciliopathies. So this has for long been like the one organelle that we've been missing in the Protein Atlas. But since it's hard to study, we didn't do it earlier. So now we set up a new imaging pipeline where we did 3D imaging and included some cilia markers so that we were really able to detect these. Can you see these little antennae that are sticking out of the cells? And then we did a targeted screen to identify proteins in the cilia in 3 different cell types of different origin to cover some different aspects of the human body. And these are not cancer cell lines. These are more primary-like cell lines, so that we can see the cilia on them. And it's pretty hard to show these images on a big screen like this, but you can see the cilia here, you can see the markers, and we could even annotate the protein to be in the tip of the cilia, the base of the cilia, the basal body, or in the transition zone. So we could use machine learning to quantify the protein expression pattern along the cilium and could demonstrate, for example, that the proteins in the different compartments of the cilium perform very different functions. For example, you have autophosphorylation happening in the tip, for example. So that is, of course, nice, and it's really, from a spatial perspective, it's cool to see that the cell is really like partitioning functions even at those small scales. And in total, we found about 700 human proteins in cilia and 91 new proteins that were never described to be in the cilia before. And what is also interesting is that 90% of these proteins were also found elsewhere in the cell. So it's really not cilia-exclusive proteins as you have in many other organelles. These are really proteins that perform functions elsewhere, and when needed, they also go to the cilium. And we know the cilium is a hub for sensory signaling pathways. What is nice is that we could also find not only these kind of receptors for signals, but a lot of the downstream signaling components were also present in the cilium. So this kind of changes the notion of the cilium from only a sensor to maybe even a little computing device or something that not only senses, but also integrates the signals before it passes it further. So there's multiple steps where you can kind of filter these signals. And for this talk, I thought particularly, did we find any enzymes? Did we find signs of metabolic activity in the cilium? So pretty little is known about metabolic and enzyme activities in the cilium, but of course it's known that the lipids are very important, for example, for hedgehog signaling. And we could find multiple enzymes in the cilium, for example, the ones shown here. So this at least suggests that there is some local production of lipids in the cilium and that the cilium itself might be able to change its lipid composition in order to adjust to certain signaling environments. But of course, more studies is needed to confirm this. And what was interesting with the cilium also from our perspective compared to many other organelles is the very close link to this class of diseases called ciliopathies. And of course, we found many known ciliopathy proteins there, as should, but we also could confirm that GPR75 is localized to the cilium, which is mutated and has associations to obesity. We found many proteins, and almost 10, that have been linked to autism. And we also found a potential novel ciliopathy gene. And this was a fantastic collaboration with clinical genomics at the Karolinska Hospital in Sweden, where we investigated their whole genome sequencing data that for patients that had a suspected ciliopathy but could not be confirmed with a regular kind of panel. So we identified a boy here that had a stop codon in CREB3, which causes CREB3 to not localize to the cilium anymore. And we, for the first time, showed that CREB3 is actually on the cilium. So this would suggest that CREB3 can also give rise to ciliopathies, for example. So again, this is very basic biological data. We're like putting out compendiums, atlases, with well, data that we hope that people can use. And again, here, this was published last year, the Cilia Atlas, and then we have landing pages and all the data in the Protein Atlas. So we hope you make good use of it. But now I will shift gears. So we generate a lot of data at scale, and it's interesting to identify proteins of interest and studying them a bit deeper. But we're really interested at the systems level study of cells. So big part of my lab is computational, and we think a lot about, for example, can we use AI to build whole proteome spatial cell models? Well, the first thing we want to do, if we want to do that, is of course to be able to interpret the images that we're generating. So I want to present a model that we call subcell that we developed pretty recently. It's a proteome-guided vision transformer model. So our hypothesis was that there's a lot of nice dino models, image recognition models out there, but they're all kind of trained in a brute force way. And we think that there's a smarter way to do it. So we thought that we could make use of the fact that we have measured most human proteins in the Protein Atlas. So we have a very high pattern diversity, whereas most other data sets have more data but less pattern diversity. And we set up this multitask learning framework, and I just want to point out one thing. So it's a classical masked autoencoder vision transformer. What is important is this protein-specific loss. So we make this assumption that localization is conserved in our cells. So if we take protein A in cell type X, it's more likely that the pattern of protein A in cell type X will be similar to the pattern of protein A in cell type Y. So they are supposed to be more similar than to protein B, for example. So we're kind of encoding that into the loss function, so that the idea was that the model should be able to recognize patterns even in cells that look visually, morphologically very different. And that actually turned out to help the model a lot, prevent class collapse. And in the end, it's a very powerful model. I'm just going to summarize it a little bit here. We can use the model without any fine-tuning to do many different tasks. Classify patterns in our own images, of course. Classify patterns in other data sets, like OpenCell or Allen cell. We can reconstruct a cell cycle in a yeast image data set without any fine-tuning. We can classify mechanism of action in drug in the JUMP data set, in drug-perturbed cells. And all of this, so in general, this is a powerful model. And again, we think that the reason for why this model is so powerful, and even better than the model that was trained on the yeast images, for example, is because we're making this assumption about the pattern diversity and how patterns are conserved. And I'll get back to that. I do want to point out that what is nice with preprints is that even though this is just preprinted, now some other people already tested our model and published that and show that it's better than existing models. And especially this plot is cool because we trained the size of the circle corresponds to the size of the training data set. This is the data we trained on. This is the data they trained on. And our model beat their model. So it kind of speaks for thinking about these clever biological aspects that you can encode in your model training. So hopefully we also get our paper published sometime. But it's nice to see that others are using it. And if we inspect so this is a vision transformer. So it has a lot of attention heads in it. So when we say that we believe that the model has learned to pay attention to proteins and regions in the cells that are relevant for biological functions, because proteins execute function, we wanted to inspect the attention heads and see what regions in the images do the model actually pay attention to. So these are some example images, and then these are in black. If a pixel is shown in black, it means that the model is paying attention to this pixel. And you can see that these different attention heads have almost specialized to different organelles, to pay attention to different organelles. Attention head 4 looks at the nucleoli. Attention head 3 looks at some regions outside of the nucleus. Attention head 2 always looks at the part of the cell where you don't have the protein of interest, and so on. And the model never pays any attention to the region outside of the cell, despite that there's no segmentation needed. So I think that this is the reason that the model is performing very robustly, because it has learned to pay attention to biologically relevant regions. So try it if you're using with microscope images. So what can we do with this model? We can take all the images in the protein atlas and bring it into a joint embedding space and a UMAP here, but instead of single-cell sequencing data, which you usually look at in UMAP plots, this is 14,000 human proteins. And you can see if we color it according to their subcellular localization, we have a nice separation. So now we're at a point that we've taken all our images to an embedding space where we can think about integrating it with other types of data. I still have some time. Okay. And I will build further on that soon. Sorry, this is like a smorgasbord of everything we're doing. A question that I often get, so I'm going to preempt that question, is if we look at these embeddings, how do they correspond to protein language model embeddings of protein sequence, for example? And actually, they don't correspond very well at all. So at the top here, we have all these proteins clustered, the subcell embedding, and then we do just a hierarchical clustering to different levels. This is ESM embeddings of the same human proteins clustered. And then we take the subcell colors and show it on the ESM embedding and vice versa. And you can see that it's all very mixed. So it seems like these embeddings are actually holding quite different information, which also means that there might be value in combining them. So we wanted to test this. So we built a multimodal embedding of protein sequence and spatial localization. And then we tested that embedding to use it to predict protein-protein interactions. And it shows that this multimodal embedding is far superior than any of the other embeddings alone when it comes to predicting protein-protein interactions. And of course, it makes sense, because knowing where the protein is very important for trying to understand who it might interact with. And it becomes even more interesting when we look at paralogs. So paralogs are genes that were duplicated at some point in time. They're often the sequence is very, very similar. Some of them remain functionally similar. Some of them diverge and get different functions. So what we can show is that when we use the spatial encoding, we can separate functionally divergent paralogs. So this speaks to that the cell might use space as an axis to specialize function over sequence at some point. So it's something that we're investigating more. But multimodal embeddings, or maybe another conclusion could be that the next generation of protein language models should maybe be improved so that they're able to reason about spatial location also. Okay, so can we then use these embeddings to build spatial cell models? And again, I talked about the value of protein-protein interaction data. In a way, it's also spatial data, because protein-protein interactions measure your local spatial environment, whereas our images measures the global spatial environment. So we built this computational framework. This has been a fantastic collaboration with Trey Eidecker at UCSD to integrate these data types. And again, bring image embeddings and protein-protein interaction embeddings into a joint embedding space. And then we can take that joint embedding space and basically do a hierarchical clustering. And we end up with a map that looks like this. And this is kind of your gene ontology cellular component map. You have the cell on top, and then you have the nucleus, the cytoplasm, nucleoli, and mitochondria, and it trickles down. But the difference is that the gene ontology map was man-made, often with the assumption of one gene, one protein, one function. But this is data-driven. So we wanted to also show what you can do with maps like this. So for example, you can pick whatever favorite complex that you have, like the RAG-regulator complex here, which it's a known complex, but we found 2 more components in it, in this cluster. And then we worked with Andrei Shali at UCSF, and he did his integrated modeling to show that these 2 new proteins that we associated to these clusters can actually dock to the RAG-regulator complex. So we can use this map to guide structural modeling. You can also use this map like you use Go, for example, to do gene set enrichment analysis and look for GWAS mutations and see how they fall on systems instead of genes, for example, and see if you get better power that way. So this is how we map the big data set of mutations in pediatric cancers onto these functional cellular systems, if you will. So I'm not going to go into the details anymore, but there is this computational framework of how you can build these integrated cell maps. And we showcased a lot of different applications in the paper, but I do want to point out that we were able to annotate novel protein function for 46 proteins in this paper, and also suggest novel secondary functions of proteins for almost 1,000 proteins. Of course, it has to be experimentally verified, but I still think there's so much to uncover when it comes to secondary functions of multilocalizing proteins. So for the last couple of minutes here, it's going to get even crazier. This is nice, but wouldn't it be even better if we can bring it back to the image space? So what can we do to generate images? Well, we can use generative modeling. So can we train a generative model to generate microscope images of where proteins are? And we've trained a diffusion model for protein microscope images. We call it ProtVL, proteome-wide virtual labeling. It's a deep generative model based on latent diffusion design with some tricks to it. So we can generate high-resolution, realistic protein fluorescent images based on these landmarks that I showed you in the beginning, the DOPI, the microtubules, and the endoplasmic reticulum. They are commonly used as reference markers. But the nice thing here is that we're not limited to the number of fluorescent channels that you can measure in a microscope. We can input these reference markers and output all human proteins, images of all human proteins, for example. So here are some example images of generated stains for proteins in all the different organelles, in some different cell lines, just to show that it works robustly across cell lines. It works robustly across all organelles. We've also compared it to some of the other models that there's been 2 papers, early models pointing in this direction. So our model is outperforming both of them. And this is the FID score, which is a score that the lower the better. And it basically measures the global characteristics of an image. But we have many different metrics in the preprint that soon will be out. And what is also very cool is that we tested image generation on a completely new cell line that was not included in the training data set, and also drug-perturbed cells. And it actually performs pretty well. So as long as your morphology is within the morphology space of the 40 cell lines it was trained on, it seems to be working pretty okay. Of course, it's still simulated images, so keep that in mind. But ultimately, I think this presents a step towards fully generative virtual cell models by enabling the simulation of the entire proteome in silico. So we've generated the first ever proteome-wide image of a cell, and we've generated 30 million synthetic cell images that we'll make available to the community, so representing all of these proteins in 200 cells from 12 different human cell lines that we hope that other people can use to compute from. And this preprint will be out next week, and I'm going to save some of what we've spent, I think, the last year on is really, again, not just generating images and saying, hey, nice, this is a lot of images, but actually computing off from these images and show that there's actual biological value in understanding biology from these simulated images. So we can, for example, use it to infer cell states. We can use it to build hierarchical models of cells. We can use it to do a spatial gene set enrichment. So if you have a list of 2,000 proteins you're interested in, we simulate those images, and then we do pixel level clustering, and we show your gene set enrichments in an image. And you can see where different biological processes happens, for example. So please, it'll be out next week. Play with it. Try it out. And with that, I also want to end on a note that at Stanford, where I am right now, there's a lot of talk about virtual cell models, and it's often, I think, it's a very lofty goal and something that I guess biologists have been wanting to do for a very long time. And very often there's a lot of focus on single cell sequencing data and how you can scale that to build better models. And I just want to say that I think the key challenge will be to connect the scales, as we already heard. As biological systems are hierarchical, and I do believe that proteins will be essential to connect the scales. These models need to be spatially resolved. They need to focus on proteins and not only RNAs. And there's a lot of work left to be done here, but it's a fun field at the moment, too. And in case you're interested in protein subcellular localization, we had a review out at the top there just a few weeks ago. There's on just protein subcellular localization as a driver of functional diversity. And then we have a paired review that is coming out in a couple of weeks where we talk about technologies to measure and modulate protein localization. There's some very interesting druggable approaches that are coming, not from our labs, but from others. And also, we have a benchmark paper where we compare protein language models and their ability to predict the localization of proteins, and they all perform pretty terribly. So it's very much an unsolved problem. So go at it too. That one will be out soon as well. So thank you. Happy to take any questions. And this is a fantastic team. This is both the Swedish and the Stanford part of the lab when we met together last year. And thanks to all the funders too. Thank you.
Thank you so much, Emma. That's really amazing work that you do, and thanks for sharing all that. And I think we have some questions.
Hi, here. Here.
Oh, yes.
Thanks for the talk. And I'm from here. I'm a PhD student. I've been working on the string database a lot. I'm one of the core members. And we are, like in my very first project in my PhD, we created the string network embeddings and use that to predict the localizations. And we found that for some of the proteins that are in multiple localizations, the string embeddings is even better than the sequence embeddings, because it provides you the pathway information. Like if proteins are in the same pathway, they will probably in the same localization. So I mean, wondering if you are for the future project, you are considering having the prior knowledge in your models.
That's a great question, and string is fantastic. I think so too. It holds a lot of information, and it's also been curated to really be reliable. We do the ProtVL model that I showed now can only inpaint the proteins it was trained on, so it doesn't generalize to new proteins. But we would love to explore that, and then I think we also have to incorporate something like string networks as a condition, and maybe even transcriptomes as a condition too.
And
yes.
I'm happy to be a collaborator if you want.
Would love to. Thank you.
Okay, we have a question here after that. Yeah, please.
Okay, thank you. Thanks a lot for a super nice talk. Yeah, my question is mostly, most of the data that we have on adipocytes, let's say, in this space is on single nuclei. But you show that the interesting stuff happens at the pathways and the cytoplasm, the mitochondria, and we are missing it. So my main question is, how can we bring your data, your embeddings, etc., to boost the power of the single RNA-seq, let's say?
Yeah, I think it can be used basically from these generated cell images. You can more or less build a network that is it's not string, it's not direct protein interaction networks, but you can build proximity networks, for example, for your cell type of interest. Which, because string isn't cell type resolved, so that would be one of the limitations where you might be able to use this. So I think you can use it in similar ways as string and those types of data to mainly denoise RNA-seq data or show the right paths, for example, to function. So that's how I would use it.
Thank you.
Okay, we have a question down here.
Mine was just because you talked about cells, but you didn't tell us which cell lines you're using. So what kind of cell lines are you using, and is there one that works better than others?
Yeah, very good question. We use a panel of like 60 cell lines. Many of them are cancer cell lines, but now we're entering a new phase of perturbations. So now we're not going to do any cancer cell lines. We're going to try to stick with more normal cells where the genotype is also normal. A lot of imaging studies is done in a cell line called U2OS, which is often also used to study DNA damage. It's not great in any other way than it's good for imaging. It doesn't move fast, and it's very flat, and it displays its organelles nicely. So it's more technical considerations like that than actual, like now we're doing a lot of work in stem cells, and it's a lot harder because they clump together, and you have to really, like your image segmentation and recognition models have to be much better for that. But then, of course, we have the tissue. The reason we haven't pushed it so much into more normal cell types is also that we have the tissue atlas. Even though we don't have the same resolution, we can confirm a lot of the findings in the tissue images.
Hi, thank you. Absolutely amazing work. Thank you so much for sharing. I was wondering, in line with cell cycle and metabolism, how is circadian rhythm and changes with that reflected in the atlas?
Yes, so the U2S cell line is one of the few cell lines that actually has a maintained circadian rhythm, but we haven't synchronized the cells. So the cells, like we have, all proteins are imaged in the U2S cell line, but they're non-synchronized when it comes to the circadian rhythm. when we do look for known circadian proteins, we don't see a lot of variation in our data set. So I don't think it's the right data set to see it. And also, if we look in the tissue images, for most of the proteins where we see single cell variability, you see single cell variability in tissues too. And if it was circadian, then maybe you wouldn't see that because the tissue was acquired at one time point. But what is more interesting in the tissues is that we might see variability in the cell line, but then we only see variability in one or two tissue types, but not all the others. It becomes even more complex when we look there. Yeah, but it's a very unexplored angle, the circadian, like exploring the protein atlas from a circadian perspective might be a fun project.
Okay, so I'm Henrik Mulder. I'm from Lund University, and I'm sitting up here. This was a very impressive and very beautiful presentation. So I have a very basic question. So I trained as a histologist in the 1990s, and we were subject to very rigorous control that the antibodies that we were using, that they were specific, not cross-reactive. So when one has the ambition level that you have working at your scale, how do you control that the antibodies are actually showing what you think they're showing?
Yeah, that's a very good question. So in the protein atlas, we use different ways to validate the antibodies, and then we also have different reliability scores assigned to each assay for each antibody. So when we validate antibodies, the best would be, for example, a knockout, and we can show that the signal disappears, or a pull down, and we detect with mass spec that it's binding that right protein. But we also know that antibodies are kind of assay and context dependent. So that doesn't mean that the antibody will bind the right protein in all contexts. So you always have to take it with a grain of salt. But we have these 4 levels of reliability. So to reach that highest level, we have to have either knockout verification, mass spec verification, or this is, I think, our favorite approach because it's been most pragmatic. If we generate 2 antibodies for the same protein, but they bind different epitopes, and they show the same pattern in a sample, we also consider it validated. But we're constantly curating the atlas, and if you tell us that something is wrong, we're probably going to trust you because we can't be experts in everything. And I would estimate that there's probably like 10% noise in the cell atlas still.
This sounds excellent. This is exactly how one should do it. But is it really possible to do that for 25,000 antibodies?
Well, we've made 25 is the ones that we've passed. We have made 60,000 antibodies, so the majority have been failed. So the first thing we do when we produce the antibody, we purify it on the antigen. So of course, if we don't get any antibody, it didn't even bind the antigen. But then we do a protein array also, and that's the only hard fail. If the antibody shows cross-reactivity to any other protein in a simple protein array format, then it's never going to work more complex sample, and then we fail it straight off. So there's a ton of data that is kind of hidden in the protein atlas, which we don't believe in.
Such a lot of work that is never really shown in these papers.
Yeah,
fantastic.
exactly.
Okay, so with that, I think we thank you once again, Emma. Thank you so much.
Thank you.
Fantastic.